Okay, so good afternoon, everyone, and I welcome all of you to this another edition of the NSF Wednesday Colloquium. And it's a pleasure for me to have one of my colleagues in the Institute, Professor Shubha Tole, who is a very accomplished neuroscientist, give the colloquium today. So before I formally introduce Shubha, although for the rest of this institute, of course, she needs no introduction, but for the broader audience who are listening to us today, I will do that. So before I actually do that, I just wanted to tell a few words about what the Wednesday Colloquium stands for the institute as a whole. It's actually one of the most brilliant ideas of our founding director, Professor Homi Bhabha, that has kept this institute connected. That every Wednesday at 4 p.m., scientists from all walks within the institute, whether it be a physicist, chemist, or a biologist, they all get together and sit together to listen to an expert in the scientific community from any of the fields or from any of the branches, sub-branches, talk about the latest and the contemporary developments that excite the people in the field. Sort of the entire colloquium is given in such a way that the challenges of that particular subfield or subtopic is enumerated for a broader audience. Today is no different. Professor Shubha Tole has kindly agreed to give a colloquium after almost 14 years, as she tells me. Actually, it's kind of interesting that when I joined, she was one of the most helpful characters to us, at least when I joined the institute. But I just wanted to say that she has had, she is a neuroscientist by training. And one of the most important works that she has done in this particular field of neuroscience is that she has discovered a very important gene, LHX2, which is pretty crucial in the formation of hippocampus, amygdala, and the cortex of the brain. And Shubha, she's of course, for her fantastic work, has been awarded with the topmost prizes, and the latest one is the Infosys Prize, which she won in the category of life sciences in 2014. Without further ado, I would like Shubha to proceed with her colloquium and give us a basic a round of a trip to how the brain functions and the circuits of sensation and how we perceive the world. Shubha?
Thank you, JD. Very, very kind of you. Am I audible?
Yes, yes.
Yeah, and screen works. Okay, well, welcome everyone to a trip through Alice in Wonderland's rabbit hole almost. The brain is the most complex structure we know. What you see before you is diffusion tensor imaging of all of the nerves in the brain, all of the nerves pathways. Okay, this riot of color, false colored for directionality. This riot of color is not a model system for anything. It is, however, the structure which inspires neuroscientists and has done so for centuries as we try and unlock its secrets and how these circuits bring the entire world to us. So that's sort of the theme that holds my talk together. Now, the brain is likened to a computer. The key difference, though, of course, is it's a biological system. Every single part of it has to be created, developed in a growing embryo, along with every other part of the embryo that is growing. And then all of its components have to be wired. The circuits have to actually be directed from point A to point B, without which the brain system will not function. To sort of underscore how we are entirely dependent on our circuits, and we are entirely dependent on our biology. I have a few sort of introductory slides. Now, this audience obviously knows all of this, but these slides just drive home the point that we are completely dependent on what our five senses bring us. There isn't any other information available. And we might have all of the brain circuitry in the world, but on the one hand, there's too much information coming in. On the other hand, there is too little. We are limited. We are at the same time constrained and enabled by our circuits. So let me show you a few introductory slides that bring home how completely dependent on our biology we are. Okay. What color is the flower is the first question. Okay, now, I mean, yes, of course, we can all see color. Some of us have variations of color perception. That's fine. But we all know that we see in a tiny, tiny, tiny bandwidth of the entire electromagnetic spectrum. I mean, our photoreceptors, our biology constrains us that way. So what color is the flower really depends on who's looking, right? So here, this photographer has taken to photographing things in ultraviolet light. Now, we can't see in ultraviolet light, even if he photographs it that way, so it's false colored for us. And the point is, right, that bees, okay, do see ultraviolet. So what color is the flower depends on who's looking. Now, we do not know whether the bee sees red and white. Like I said, it's false colored for us. But the point is that there are non-uniform pigments or structures in these petals that make the overall percept relevant to an insect that is trying to find the center. You can see both of these actually have bullseye-like patterns, okay, regardless of what color we see right now, or the bee does. So the point is what color something is just depends really on who's looking, right? So here is one step into sort of underscoring that we are dependent on the limitations of our biology. Here is another. This is a beautiful Kanita illusion. I think everybody sees a white square sitting on top of what might be black circles. But as some of you may know, the white square is an entirely illusory contour. You notice if I block things out, the contour goes away here. There is actually no brightness difference. You continue to see a contour there, but blocking it out, it's gone. So this illusory contour, it's the Kanita illusion. This is a powerful illusion. It is not yet clear how this arises. Okay, it arises because we are wired up like this. And yet, these kinds of percepts are important to our lives, which I'll bring out in a little bit, and not just important to our lives, they're important to lives of every living creature. Now, a recent study just was published last month, which I found very interesting. Okay, it is known that cats will sit in small, little tiny locations. Wherever they can fit, they will sit. What Gabriella Smith at the City College of New York, I believe, wanted to test is will they sit on just squares marked in the ground, for example. So there is not actually any square hole or cavity here, but the cat will preferentially sit in a marked square as opposed to an unmarked square. And she actually designed this experiment in the pandemic, sent kits to people at home who had cats and instructions on how to conduct this experiment. And the real kicker is this. Cats will not only sit preferentially in marked squares that are marked with tape, but apparently also in Kanita squares. Okay, they will preferentially sit here where there is no square really, over the control where the little Pac-Men are oriented differently. So this tells you that this phenomenon, this percept, is a evolutionary conserved mechanism. And why shouldn't it be? Why shouldn't it be?
Can I ask a question, Shubha? Sorry. Even in the introductory slides. Is it something that the geometry of the marked space is important for the cat, or circle could also have done equally the same job?
There are many versions of Kanita illusions. So if you look them up, anything that, so many lines together will create the illusion of many ends. So triangle illusions, many straight lines like this will create the illusion of a line boundary like so. So, I mean, this is the study that was done. So I just wanted to underscore that we are really constrained and, our circuits give us this and we see this. There is no way to unsee it. Okay, why we see it, it is not actually known. Yeah, but if you look up Kanita illusion, you'll see many marvelous ones. Many. Yeah. So in fact, Geldard said some decades ago that in neuroscience, illusion is the perceptual rule and not the exception. Okay, this is a pretty dramatic statement. I'll give you one example of this. Image completion. Image completion is something our circuits have to do. They have to do it. Why do they have to do it? Because the world does not come to us in complete images. The world comes to us in partially obscured images. Almost everything we see is partially obscured. And we have to figure out, for example, that there is a Bisleri bottle behind bars here. Now, of course, we do not know the content behind these bars. Okay, behind these bars could be brightly, pink flowers. Behind these bars could be anything. Or actually, the most limited description of this is that there is a little piece with a cap and a plastic, and then there is a separate little piece with just a plastic, and then there is a separate little piece with a green, that happens to be aligned together like so. And, we think there's a whole bottle there, right? I mean, that's the very limiting description. The point is we complete images. And we need to complete images because this is a survival skill. How else would we know what's there in the world? It helps if the different parts of the image are moving together. Okay, and there are many illusions like this where random dots, if their movement is just coordinated, automatically you'll see a ball moving or something. So our brains have execute this phenomenon called Gestalt, where things are bound together. I'm not going to talk about how it works. Okay, how it works is actually one of the at the forefront of neuroscience today. I'm using these to motivate that the circuitry is important because, in some sense, how the circuitry wires up sets up the hardware that sort of runs the software. Okay, now these are survival skills. Why are they survival skills? Well, we would, if we could not complete this image, we would all be eaten alive. It's a survival skill not only for the prey, but it's also a survival skill for the predator, right? Because the predator also needs to see complete images. So, okay, I won't belabor this point, but I have one more sort of dramatic example of how we have what we have when it comes in. And when our senses compete with each other, they execute the best possible, synthesis of what it is that different senses bring in. It seems to be it's the job of the brain to say all is well and give one combined percept. Okay, and this example brings it out as well as anything else I know. This is called the McGurk Effect, and you just have to look at the video and tell me what you hear.
Ba, ba, ba. Ba, 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 ba.
So that was audible, right? Baby sounds, ba, 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 ba. Okay, now hear this one. JD, that was audible, right?
Absolutely, yes. Papa.
This one.
Ba, 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 ba.
No matter how many times I look at this, I hear ba pa pa for the first one and pa 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 for the second one. Now, it might shock people to know that it is actually the same soundtrack. Okay, it is the same soundtrack, and here I'll play it with both the videos together. You can look at one face, you can look at the other face, or you can close your eyes. When you close your eyes, you'll hear the original soundtrack, and when you look at one and the other face, the percept will change.
Ba, 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 ba.
Okay, now this is a useful skill, right? This is not just a sort of magician trick thing. By the way, this is also why ventriloquism works. Okay, our brains will hear sound coming from dispersed speakers in a theater, for example, or when a ventriloquist has a dummy, and will assign whatever is moving to the sound. Okay, so when you give a talk in a brightly lit room and the sound is coming from the speakers, you don't know where it's coming from, and the speaker will go where. The minute the speaker sees the person, automatically their percept of the source of sound becomes that person. We've all experienced this. So here is an example. If you don't mind, I won't take questions on these illusions because they are here to motivate the idea that how our brain wires is important for its function. And if the wiring diagram is not executed properly in development, then we will not have any of our abilities that are important to survival. So my interest then in research is in studying the development of the brain, not how it works, but how the circuits develop that enable it to work. Okay, so I'm going to Hello. Okay. In order to build a brain, okay, a brain doesn't build itself in isolation sitting in a bowl. A brain builds itself while there is drama. There is no other word for it. Drama happening in the whole embryo itself. And here is a video from the lab of James Smith that shows a frog embryo developing. Okay, and this is nothing short of dramatic. I'm going to, it's 30 seconds and how much changes. In reality, this process would just take a few hours or, okay. That's the egg, many, many cells. All the cells are rolling in. Inside is the yolk. And now look what happens. This forms a groove and then it rolls up on itself. And this is going to be the central nervous system, the spinal cord. This is the spinal cord happening before your eyes. And the front end of it is going to bulge out and become the brain. There you go. See the two lobes of the brain? Amazing. Huh? I'm going to play it just once more. And look at all the cell movements that have to happen. Look at the coordination, the choreography that has to happen in order for this body plan to execute. So remember this, because the second part of my talk is going to actually talk about how this axis is organized in exactly this direction and why it doesn't go orthogonal, or how is it that there's just one axis. The second part of my talk is going to really talk about this. But first, let's talk about circuitry. Okay. Even as the embryo grows, the brain is growing too. Eventually, the brain is going to have a lot of neurons. This is one neuron. It's got input wires and one output wire somewhere. Each red dot is some other neuron connecting to this one. And on average, in the human brain, it is said that there are 100 billion neurons and each makes 1,000 to 10,000 connections. So you can see the wiring problem and you can see why circuitry is extremely challenging to study. This is the original picture again. This is the result of many, many of these neurons forming the brain. This is a cartoon that shows the human cerebral cortex, and it broadly shows where the sensory modalities are mapped. Now, all of our senses, okay, our skin, touch, which I'm going to talk about, the wires from the skin come in through the spinal cord, through the hindbrain, the wires from our eyes go through the ears, and all the wires have to be collected, and they are collected into a central structure in the brain called the thalamus. Okay, in this thalamus, they're not mixed up. What would be the use of bringing in separate modalities and then mixing them up and then having to sort them out again when you connect to the cortex, right? So in the thalamus, they are separated by modality. And then this tract from the thalamus has to grow to the cortex where it spreads out, literally. So these lovely wires you see are the sensory tract spreading out, and amazingly and reliably and reproducibly, okay, in every mammal. So every mammal means a little mouse on the street, a human baby, a monkey. It is always the case that the visual cortex is back here in pink. Okay, ahead of visual is the sensory cortex, which I'm going to talk about. Auditory cortex is on the side. Taste is buried in there. So this map is reliable. How this map forms is again something I'm going to talk about in the second part of my talk. How structures are positioned where they are. But first, I'm going to talk about how the wiring gets to the cortex in the first place, how the sensory tract enters the cortex. And here's a cartoon. This is a cartoon of a mouse embryo. And here, this green blob represents the thalamus. So I'm going to usually show things only in one half of the brain, though the same thing happens in the other half as well. And this is the thalamus to the cortex pathway. This tract has to navigate through complex territory and then grow into the cortex in a modality-specific way. Because if you're not modality-specific, and in that, if you're mixed up, then your percepts are all mixed up. Okay. Here is an example of a real mouse section. And this green is where the thalamus is and where the nerves originate. And this is the bundle that now has entered the mouse cortex. So I'm going to use these terms, bundle of sensory nerves. Okay. Now, in order to perceive sensation at high resolution, okay, what would you do? If you wanted to photograph something at high resolution, you would go out and buy a multi-megapixel camera, right? So similarly, the brain allocates its resolving power to where it is needed. So the whole body is not represented in a proportional way in the brain. What would be the use of having as much sensitivity on your left toe, for example, as you have on your fingertip? Right? So if you allocate more processing space, cortical space, to any part of the body, you're going to have better resolving power in that body. The nerves from the fingertip will spread to a larger part of the cortex, so you can resolve 2 points better. Okay. So I'm going to show you now a picture of how the human body would look in proportion to how much brain territory each part gets to perceive sensation. Okay. And as you can see, what we know to be our most sensitive body parts, our lip, tongue, face area, our hands, our fingertips, they get disproportionately more cortical space than the rest of the body. Okay, and you can do this experiment with a friend. It has to be a very good friend because you'll have to take 2 toothpicks or pins and see, at what point, in their arm or their finger can they resolve 2 points apart. Okay, so it's a fun experiment to do if your friend is kind. Now, here's the same thing for a mouse. For a mouse, the whiskers act like fingers. So a mouse, before it goes through a gap, will literally whisk and sort of make sure the gap is big enough to go through. And again, similarly, for a mouse, a disproportionate part of its brain space is allocated to the whiskers, and indeed the snout and the upper jaw and lower jaw, and of course the forelimbs with which it holds things, and the rest of the body not so much. I mean, it's pretty amazing, right? And in the data I'm going to show you, I focus on this whisker to brain pathway because it's an excellent model system to study sensory innervation. The whiskers are not just random. Each whisker connects to a particular structure in the brain, and this collectively is called the barrel field. Cool classical studies have been done where if one whisker is removed, ablated, then that barrel disappears. Okay, and so it's a perfect one-to-one mapping. The pathway is not a one-step pathway. It goes from the whiskers to the hindbrain, a little relay, to the thalamus, which I mentioned, a relay center, and then to the cortex. For the purposes of talking, we're just going to talk about it whisker to brain. Okay, so what about this thalamocortical pathway, thalamus to cortex? We made an interesting discovery. Okay, but first, let me assure you, are there really barrels in the brain? Well, this term came up as a result of 2 scientists, Muesli and van der Loos in Lausanne, inspired perhaps by stacks of beer barrels. Okay, when the mouse brain or the rodent brain is flattened, okay, this outer sheet is just flattened and stained for enzyme cytochrome oxidase, it turns out that the centers of the barrels where the nerves from the sensory nerves from the thalamus come in, okay, they are enriched in cytochrome oxidase, and you can see barrels. So this is what an actual brain section looks like. These brown things are where the nerves from each whisker, via 2 relay steps, come in and form a one-to-one mapping. Okay. Well, so my former PhD student, Ashwin Shetty, examined a mouse that was mutant in one single transcription factor. It has a name, LHX2, it's just a name. We stumbled upon this transcription factor because we had found it to play fundamental roles in brain development. And indeed, we have discovered that it plays multiple distinct roles in different parts of the brain. So here's what Ashwin discovered. If LHX2 is knocked out, this gene is deleted, only in the cortex of the brain. Okay, not in the rest of the body, not in the thalamus. There's nothing wrong with the nerves coming in. But if you just remove it from the destination, here's what happens. So that's Ashwin. He's now doing a postdoc at Harvard Med School. And here is the LHX2 knockout mouse. Okay. Hide nor hair of a barrel to be seen. Now remember, we did nothing to the actual nerves coming in. Okay, those were completely normal nerves. But when they hit the cortex, for some reason, barrels could not form, did not form. They could not enter the cortex. This was a 1-week-old mouse. So we reported this. We examined it in some detail. And then we decided to go back in development to find the genesis of this problem. Okay, so now in order to study the genesis of this problem, I have to introduce a little bit about this knockout business. When I said it's knocked out only in the cortex, there is some detail. Okay, we have reagents, which are mouse lines, that can selectively knock out a gene, okay, in select time or place in the body. For example, a standard knockout, which I will talk about at the very end of my talk, is when the gene is just knocked out. It's knocked out in the sperm, in the egg, and right from day 0, the whole animal is just knocked out. Now, that's not a really good way to study what a gene does, because you would like for it to be a very selective knockout. So in this case, where is the gene knocked out? Only in the cortex. And when? At an early enough stage in the embryo, when all of the cortex is largely still proliferating. All of it is still stem cells, okay, mother cells or stem cells. Proliferating cells will give rise to neurons that no more proliferate. So the neuron that is born out of a stem cell or a mother cell, that daughter neuron is now the neuron that the animal has for the rest of the animal's life or the neuron's life, whichever ends first. Okay, so neurons don't proliferate, but the stem cells do. So this gene is knocked out early enough in the embryo in all of the dividing stem cells, which produce all of the neurons of the brain. And in this situation, we don't see barrels. So now to explore further, how did this profound defect come about? How did it arise? So here is the cartoon again of the thalamocortical pathway. And here you've seen before the green thalamus and the labeled tract. Now, how is this tract labeled? In order to label it, we had to actually develop a very difficult technique, because it's not easy to just make one particular pathway green. Okay? And in this, so this broad project is the thesis project of my student Suranjana Pal, who finished her PhD thesis recently. And this particular labeling technique was perfected by a former postdoc, Geeta Godbole, and a former master's student, Tuli Pramanik. What they did was they introduced a GFP carrying plasmid into the thalamus at an early enough stage when the thalamus was producing its neurons into the stem cells of the thalamus. And they did this by a technique called in utero electroporation. Okay, so that means, so the whole embryo is like less than my little fingernail, okay? And it's in the uterus. So actually, let me show you. There. So this covered in gauze is the mouse. You can see this is the chin area of the mouse. These are the forelimbs. It's under deep anesthesia. It feels no pain. Here is a small glass microcapillary with which DNA is being injected. This is just illumination. And then electric pulses are given that allow the plasmid to enter whichever cells are around it. So the injection has to be done very, very precisely. And this is how this tract can be labeled. And this is the technique we're going to be using. So what did they find? Now, in a normal mouse, 1 week old, these are sections of the brain, and you can see these barrels. Okay, because they are sections, you don't see all the barrels, you only see one plane of them. And here in HiMac, it's quite beautiful. Look, this.
Sorry, Shubha. Shubha, can I ask a question?
Yes,
yes. Which part of this is labeled? Like, how did so the GFP is labeled to tag to what?
It's just a GFP expressing plasmid that expresses green fluorescent protein.
That's it. Okay.
Yeah.
Okay.
So
okay.
It will just be expressed in the neuron, and it will just label all of
the neurons. Okay.
It helps if it is a membrane tag GFP, because then it labels all the fine arbors.
Sure. Okay. Thank you.
Sure. So you can see how beautiful these arbors are. Okay, these are the barrels, and these are called infrabarrels in another layer. And this is the tract coming in. I want you to note this. This is the tract coming in, because remember, when we look at the mutant, we did nothing to the tract coming in. We only mutated the gene in the cortex. Okay, and here's the mutant. Yeah, this is the comparable section, and maybe some smidgen of something here. And look, here's the tract coming in, and it looks like the nerves just haven't penetrated. Right now, this is 1 week old, but when does the tract come in? The tract actually reaches the cortex 4 or 5 days before birth. Mouse gestation is 19.5 days, and by embryonic day 15, the tract has come and entered the cortex. And this is a very interesting phenomenon. It comes and enters the cortex, and then it waits before it actually penetrates. And why does it wait before it penetrates? Because these cells up here have not been born yet. So the tract comes first and bides its time, and this is going to be central to the coming few slides. Okay. When Suranjana looked at embryonic day 15, this is the control, and here it is, the tract has entered, and you notice it has not innervated this black layer that you see on the top. It's thin because the neurons haven't yet been produced. The neurons are going to be produced from mother cells, stem cells underneath here, and they're all going to grow outwards, and the cortex is going to become thicker, but all that hasn't happened. So this tract is peacefully waiting. What happens in the mutant? This was a real surprise, okay? Look, there is no waiting. There is no nice tight bundle. There is in fact random and premature ingrowth into the cortex. And this is completely in contrast to what we see here. Okay, after birth, we see that, things have not been, maybe the cortex is hostile or something. There are different hypotheses if you look only after birth, that for some reason things couldn't innovate. In the embryo, not so. Not that they couldn't innovate. In fact, they're doing too good a job of it and doing it prematurely and incorrectly. The timing of this, the fact that it is in the embryo at day 15, presented a hypothesis to go forward, and I'm going to talk about it. But before that, I'm going to contrast this with a different mutant. Remember, I said this LHX2, we have knocked it out in all the stem cells of the cortex. Okay, and this gene acts from the cortex knockout acts from day 11. And this embryo is examined at day 15. Now, there is another genetic driver we have, okay, which can knock out a gene of interest, not in the stem cells, but in the neurons immediately as they are born. So that too acts from as early as day 11. Okay, now this is just a very dramatic view of an overshooting type of, this is definitely not normal. But here, this is the neuron knockout. When LHX2 is knocked out in neurons after they are born. And this is immediately after they are born. Okay, so we are knocking it out at day 11 still, but only when after the neurons are born. And then if you look at day 15, the tract is obedient. The neurons don't have LHX2 and still the tract is obedient. So this told us that what is likely to be happening is that there is a stem cell specific mechanism in play, and it is likely to be stem cells of a very special type of neuron that I'm now going to talk about. Okay, so here is just some quantitation. This is the control, and this is the neuron knockout, where you can see that the aberrant branching is really minimal. And here is the stem cell knockout, where there is a great deal of aberrant branching. So quantitations of multiple ends confirm this finding. Now, what is this special set of cells that immediately comes to mind when we see a problem at embryonic day 15? Okay, it's a set of cells called the subplate. The subplate can be considered as a park and hold array for incoming nerves. What do I mean by that? Here is the normal cartoon. And remember, I told you the tract doesn't innervate the cortex right away, but rather it waits. These blue dots are the subplate. And the subplate is critical in order for this tract to wait while the layers of neurons upstairs are still being born. Okay, I won't show these yellow dots again, but these are the neurons that are eventually the neurons of the cortex that need to see the sensory power, need to get connections from the sensory pathway, but they're not formed yet. So the tract waits. Now the subplate is so important that classical experiments decades ago, Carla Schatz and colleagues, when they ablated the subplate, the tract couldn't find its way. Okay, the subplate has been classically known to be important. And in fact, so important that in mutants where the subplate is misplaced, look here, it is all the way at the periphery of the brain, the tract actually goes and finds subplate cells there. Okay, this is a mutant called Reeler. This is another P35 mutant where the subplate cells are disordered and scattered all over, and the tract goes and finds them there. And these studies are now more than 2 decades old. So this has been the sort of prevailing hypothesis that the subplate is critical for these nerves to wait. And what it does is not clear. Okay. In the rest of my talk, in part of my talk, I'm going to give one insight into what the subplate may do, may provide to these incoming nerves to allow them to wait. And the reason we can answer this is because we have a mutant in which the axons don't wait, the nerves don't wait. Now, immediately after I show you these cartoons, obviously you're going to think, well, look at the subplate in your embryos. Maybe the subplate is, sort of all the way up here, which is why the neurons are, the fibers are growing all the way. So that's immediately what we did and looked. Okay, so this is a control subplate, this tight row of cells here. This is the subplate in the neuron-only knockout. Remember where the tract was just fine, no problems there, and the subplate is fine. And now in our stem cell LHX2 knockout, we expected the subplate to be either disordered or all the way up here so that it would attract the nerves, but no. Okay, it is slightly fuzzy, I admit, it is slightly not well packed. But given these kinds of findings, classical findings, wherever the subplate cell is, the neurons are going to go and the nerves are going to go and find them. This subplate is located pretty okay. It's not all the way up here so that it would attract all the nerves. So this prompted the hypothesis that it may not be the location of the subplate that's a problem, but for the first time, we seem to have uncovered a gene that is controlling key properties of these subplate neurons that they are unable to now offer something required to the nerves to make them wait, or maybe that the nerves no more recognize them. Okay, so we are now in a position to ask what property of these. Okay? And here's what we do.
Shubha, again, quick.
Yes.
In the previous slide, the images that you have in the lower panel,
Yes.
how did you do the images? How did you look at these? Especially the color.
This is in situ hybridization for messenger RNA of a gene that is expressed only in the subplate, CTGF.
Okay, so one specific marker, one specific
marker. It's just a way of asking, are the subplate cells where they are supposed
there
to be?
or not? Yeah, where are they spatially?
Yeah,
and this is also some confocal fluorescence, or not confocal, but maybe it's just the fluorescence. Just the fluorescence, bright field.
It's not fluorescence. It's just bright field. This
just
is
bright. Okay.
phosphatase reaction.
Okay, just a bright field. Okay.
Yeah, it's like old classical stuff now. Yeah, it's okay. So thank you. Please ask. Okay, it's important. All right. So, are the molecular properties of the subplate disordered? So for this, Suranjana microdissected the subplate. Okay, this is like a 1 millimeter strip that she had to take out. And she microdissected it from the stem cell LHX2 knockout and its control, its littermate control, and the neuron knockout and its littermate control. And basically, when you send that tissue, when you extract the RNA and send that RNA for RNA-seq, you get a whole list of genes that are dysregulated, either up or down regulated. Okay, so in each one, which genes are up and down regulated, and then she compared these two data sets. Okay, so these are just numbers, but this itself is an interesting surprise, right? So both of them are subplate cells that do not have LHX2. This yellow ball is subplate cells that lost LHX2 in their stem cells, and the purple ball is subplate cells that lost LHX2 after the neuron was born. It's not a whole lot of time difference. It's just a stage of the cell cycle difference in some sense. But look as to how little overlap there is, right? So this immediately tells you that LHX2's role can be completely different just as soon as the cell is born versus in the stem cell. So obviously, since we see our problem, our profuse innovation in this, we looked at these 328 genes with some carefulness. And I'm not going to belabor the point. We got many categories of genes. Now, we focused on ion channels and transporters because we felt that the incredibly profuse phenotype that we see is unlikely to be explained by just one or the other, say, axon guidance or signaling molecule. And when these act in concert, they are unlikely to all create the same type of effect because all of these have slightly different functions. However, ion channels and transporters and so on, when they act in concert, they will eventually affect the electric properties of the cell. And that is one thing that we can have a readout for. So, given the large numbers of these genes that we found to be dysregulated in the stem cell knockout, we decided to examine, are these subplate cells electrically normal? Okay, marker-wise, we had 1 or 2 markers showing they were normal, but what about the electrical properties? The kaam ki baat, as it were. So, Suranjana took a trip to NCBS, Suranjana and her mice, hosted kindly by Upi Bhalla's lab, and worked very closely with Upi's PhD student, Dipanjali Dwivedi, and recorded from these extremely slippery, tiny embryonic and subplate cells. First, spontaneous activity. To our surprise. So let me walk you through this one example here. This is a mutant neuron, and the blue is a control neuron. A control neuron will normally exhibit action potentials. Each spike is an action potential, a rapid depolarization and a repolarization of the membrane potential, and will spontaneously do it, okay, at embryonic day 15. This example of a mutant neuron, not very active. And if we look at a larger population, while in control only 36% of the cells were silent, in the mutant a whopping 88% of the subplate cells they recorded from were electrically silent. Now this is pretty dramatic, okay? Because subplate cells are known to make electrical synapses with incoming thalamic nerves, and they are known to make electric contacts with, in fact, the migrating neurons that are going to later migrate and sit upstairs. Remember those yellow balls? Electrical activity of the subplate cells has been shown to be important for that. Okay, the migrating cells that the yellow balls as they sit on top. Electrical activity has not at all been examined for the incoming, for the waiting period of the thalamocortical pathway. Now, they looked at not only spontaneous activity, but they looked at evoked activity. Evoked means injecting current. Okay, so current is injected in steps of 5 picoamps into the cell. And let me just walk you through this. So this blue is a trace of one neuron, and each line is in response to one step of increasing 5 picoamps. And you can see that at a certain point, the cell reaches threshold and starts exhibiting action potential spikes. Okay, so this is evoked activity. When current is injected at a certain level, you will see spikes. This particular mutant neuron, no matter how much current is injected, not a single spike. And as a population, you can see that as a population, they are basically reluctant to fire. They are reluctant to be electrically active. So this is an electrically.
Can I ask a question, Shomita?
Yes.
So why is it that, how is it that the 20% or 12% or whatever is still active? Why is it not 100% not active?
This is biology. The subplate,
yeah, I know, but the
subplate is diverse. People are only now understanding how diverse the subplate is. Okay, so that's one answer. The other answer I can give you is that some fraction of the subplate is already born before the mutation kicked in the stem cells. So I already told you it doesn't do any good to delete LHX2 after the cell is born. And the driver we use starts acting from embryonic day 11 and a half. The subplate starts getting born in a little bit from 10 and a half onwards. Okay. So some cells may have been born that year. I mean.
Okay. Got it.
Okay. So we have now shown that loss of LHX2 prevents the thalamocortical nerves from behaving normally. They overshoot the subplate as if it's not there. And loss of LHX2 results in an electrically muted subplate. We haven't tied the two together causally, because in order to do that, we would have to somehow now energize and make electrically active this mutant subplate. There are ways of doing that, but that's sort of the next ball game. But so this is where the story is at. We have found for the first time, so I'm going to summarize this part because I think I'm running really late. We have found for the first time a mechanism that operates in stem cells of the cortex. Okay, this is the first mechanism identified in stem cells of the subplate. That causes premature growth of sensory nerves without mispositioning the subplate or anything, and also causes the subplate neurons to be electrically silent. So that summarizes the sort of circuitry and sensory guidance portion of my talk. I'm going to now take a step back and ask a more fundamental question. How do brain structures become positioned correctly in the first place? How do for example, when the nerves come in here, how do they know that this is where they have to make barrels and not somewhere else? We were privileged to obtaining a fundamental insight into brain patterning, how structures are formed where they are, with reference to not the barrels, but a different structure in the brain called the hippocampus. Okay, this here shown in yellow, the hippocampus is the recorder of memories. Okay, and I will now move to this smaller section of my talk. In order to introduce what we found, I'm going to take a step back and into how the body plan is patterned in the first place. Remember that video I showed you where the whole embryo is doing this complex movement and the whole neural tube is forming? How does it know to form just one neural tube and not many? How does it know to form it there and not somewhere else? Okay, these are very fundamental patterning blueprint questions. And a fantastic Nobel Prize winning insight into this came from the experiments of Spemann and Mangold, where they found the primary organizer of the embryo. Okay, that's literally how the primary organizer. What did they do? They used amphibian eggs, newt, I think, salamander or newt. These are frog eggs. I'm just showing them to you as an example. But the point is, when all that movement is happening, there is a small structure in this moving, morphing embryo that serves as an organizer, And they found this because if they removed it from one embryo and put an extra one in a different embryo, giving that embryo two of these structures, okay, so here is a Xenopus larva fully formed. Okay, so in this particular experimental animal, which had two of these putative organizers, they were able to duplicate the whole body axis. Okay, so essentially this organizer mechanism then, they basically opened up how complexity is generated from simple organization. You have a uniform sheet of cells, an organizer like a lighthouse in one side of them, and by emitting signaling molecules at different distances from this organizer, there can be different readouts, and that's how the entire embryo's axis is patterned. And indeed, after this, a few secondary organizers were identified in different parts of the body and the brain, and we were privileged to find the organizer for the hippocampus. So now hippocampus, the name means seahorse, and this is a dissected human hippocampus, and you can kind of see why people named it that. And I have to introduce and thank the amazing team that led us to this discovery. Lakshmi, Vishakha, Satyadeep, Satyaki, young master's student, Nandini, and Ben. And the funding, this was actually the dream with which I started my lab. It took us 8 years to discover this, and it's been quite a trip, so I will not delay and tell you about it. Here's what we did. Okay, what I'm going to show you is data that supports the conclusion in these cartoons. This is the sheet of tissue, okay, in the middle of the brain, and at the end of the sheet is a structure called the hem that we discovered to contain signaling molecules that are necessary for the hippocampus. Okay, so what we did was we created a mosaic embryo with multiple hems. Now, normally, next to each hem develops a hippocampus. This blue and pink are all different parts of the hippocampus. And what I will show you is that in this mosaic, next to each hem also develops pink and blue, and not in a randomly messy way, but actually elaborated, structured. Okay, here's the data. How did we make multiple hems? Turns out LHX2. LHX2 at a very early stage in the brain, much before there are, nerves and thalamocorticals and so on. LHX2 is expressed in the primordium of the entire cortex. This is again mRNA in situ. And you can see how it's not expressed in this tiny structure at the tip. And this tiny structure expresses Wnt genes. Okay, it also expresses BMP genes. These are two major signaling families. Wnt genes are very important in the body. They are a powerful signaling family. I will talk exclusively of what is called the canonical Wnt pathway. Wnt genes, when dysregulated, can cause cancers, can cause multiple developmental defects. And here now is a Wnt-rich structure sitting right next to where the hippocampus would form. The first study from my lab, a master's student, Sharda Bulchand, discovered that when you look at an LHX2 mutant brain, okay, this is a full body mutant from day 0. That's what we had. Okay, just the whole thing is LHX2 mutant. Turns out that there is no cortex or hippocampus, and that everything has now differentiated into hem. So there's an expanded hem here. And we took advantage of this finding, okay, because we basically hypothesized that maybe all of these cells need LHX2 in order to become cortex and hippocampus, and without them, this hem fate appears. So we thought, what would happen if we created a salt and pepper embryo with, LHX2 mutant and normal cells? Because see here, there is a great big signaling center, but nobody for it to respond to. So if we created salt and pepper, we would have signaler, responder, and then we could do the experiment that we wanted. So in order to do that, so we did not have this expertise, and many, many different colleagues helped us from all over the country. So we are going to show you now mosaic embryos where the black cells just don't have GFP, so they're not fluorescing, and the green cells have GFP. This here is the inner cell mass, which eventually forms the whole embryo. And the green cells will be either normal or mutant, and then that way we have control mosaics and mutant mosaics. Okay, so here is a control mosaic where both black and green cells are normal. And you can see this nice salt and pepper, and you can see the cortex is normal. This is a marker for the cortex. You can see the hem is normal. It's a marker for the hem. Now when the green cells are mutant, okay, you can see there are multiple hems, and these are actually the same sections. I'm going to overlay them. You can look at this triangle here, for example, or this square. Basically, wherever LHX2 is gone, there the clump of cells becomes hemmed. So now we have a situation in which we can ask, hey, what do extra hems do? And here's what they do. They make multiple hippocampi. Okay, so this is a little older embryo. This is a little baby hippocampus. And this is the hem, which dwindles with age. And here is a hippocampal marker prox1. Okay, a normal hippocampus beginning to form. Here is the mosaic. Each green clump is mutant for LHX2 and therefore becomes a hem. And next to each hem develops a new prox1 cluster. I'm going to show you one example of a slightly older embryo. Ignore this dark patch, which is actually the thalamus, which is not important for this section, but it expresses this gene. Look at this V shape, this C shape. These are parts of the hippocampus. This is the normal. Here is the mosaic with multiple hems. And again, ignore this dark thalamus, but look here, one hippocampus, maybe two, maybe three, maybe four. So they don't all form in the same orientation, because it's a 3D structure. So depending on how they form, and when we cut them in one plane, we see all these effects, but they are actually patterned. We've looked with multiple markers. It's not just a hodgepodge of cells. We have de novo hippocampal patterning here. So we've discovered then a organizer in the brain that is sufficient to create an ectopic hippocampus. And that's the summary for this section. I have just a small section left. Okay, so what have we done? We've pushed back the question, right? Why is the hippocampus where it is? The hippocampus is where it is because the hem is where it is. So then, of course, why is the hem where it is? Right, that's the next open question. I'm not going to show you data for that. We've made some progress. There are other fundamental transcription factors which collaborate with LHX2 in regulating the position of the hem. And I'm not going to show you that data, but obviously that's the next question that we have to answer. What positions the hem? What I'm going to show you is 2 slides that ask a different open question, which has been bugging me for years and years. Directionality. Why doesn't the hippocampus form on the other side of the hem? Okay, what is there on the other side of the hem? Okay, this cartoon, I guess you're now familiar with this structure. This green is the hem. This would be the hippocampus. On the other side of the hem is a structure called the choroid plexus. The choroid plexus secretes cerebrospinal fluid. Okay, it's CSF. It's like the serum plasma for the brain. It carries with it protein goodies. It supports nourishes the brain. All of the molecules in the CSF are secreted by the choroid plexus, and the choroid plexus itself arises from the hem. Okay, I use the word arises. So the hem does not produce the hippocampus. The hem induces the hippocampus, which means that the cells that actually form the hippocampus are different, and the hem is merely signaling them like a lighthouse to become hippocampus. As opposed to the choroid plexus is literally produced by the hem like toothpaste. So if the hem was a tube of toothpaste, the choroid plexus would be the paste being squeezed out. Okay. Now, given that the hem has all these Wnt signals in it, okay, and in work that I didn't show you, we know that Wnt signals are what the hippocampus are what induce the hippocampus. Why don't the Wnt signals operate in the other direction? Okay. And this is a question that was asked by this wonderful team, Mallika Chatterjee, a former postdoc who is now a faculty member at Amity University in Noida, and Arpan and Varun, who are current PhD students. So they used a cool genetic tool that we have. So now you folks know why we have this animal house with so many different strains of animals in it. Okay, this is a strain that can drive a gene of interest only in the hem and choroid plexus. And what they did was they drove a version of the Wnt downstream factor that effectively hyperactivates canonical Wnt signaling. A persistent and perpetual activation of canonical Wnt signaling can be achieved in our location of interest. And here they did it in the hem and the choroid plexus. Okay. And now this is a little bit of a lo and behold kind of finding. The lo and behold finding is this, okay? They dissected out choroid plexus and hippocampus from both the normal mouse and this persistent perpetual canonical Wnt activation brain. And then they put it through RNA sequencing. And here is a PCA analysis, which shows very clearly that the control choroid plexus is hanging out here far from the control hippocampus and even the mutant hippocampus, as well it should. But look where the mutant choroid plexus is hanging. Okay, this hyperactivated Wnt choroid plexus is hanging out closer to the mutant choroid plexus is hanging out closer to the hippocampus. Okay, what does this mean? They looked at the whole RNA-seq data set and saw indeed that a whole bunch of choroid-enriched genes that are normally enriched in the choroid are downregulated, okay, in the mutant, and a whole bunch of hippocampus-enriched genes are actually upregulated. Okay, is this really the case? You know, aankho dekha hal? Here are some examples, 3 examples. So if you look at this white outline, the normal choroid plexus never expresses neuronal stem cell marker. Okay, this is PAC6 for people who want to know. Never expresses PROX1, the same marker we used for the multiple hippocampus thing. Never expresses beta tubulin, which is the marker specific to all neurons. And look here, the Wnt activated choroid plexus has this and this and this happily, and here's the quantitation. You can see the control numbers for these markers are all at 0. And in the mutant, you now see expression of neuronal markers. So we're effectively converting the choroid plexus into a hippocampus by hyperactivating the Wnt pathway in it, which leads to the hypothesis that normally in the choroid plexus, there must be internal damping mechanisms that prevent it from responding to this Wnt signal coming in. And in some sense, it protects itself by maintaining just the right amount of Wnt signaling. Wnt is necessary for choroid plexus, but some level of it. Too much of it and it becomes hippocampus. So this tells us that the normal choroid plexus must have these Wnt damping mechanisms in it. And that's one line of future exploration. But what I'm going to close with is a cool direction in which we took this project. We collaborated with Orly Reiner at the Weizmann, and my student Arpan Paricha spent almost a year in her lab learning to make human embryonic stem cell derived organoids. Okay, an organoid is, you can think of it as a mini brain in a dish. These stem cells are, here's Arpan. Stem cells are aggregated into little balls and then cultured. So for 18 days, the original aggregation protocol. And after 18 days, a set of key signaling factors have to be added, goodies, in order to achieve choroid differentiation. These goodies contain factors that essentially simulate what the hem normally contains. Remember I said the hem contains Wnt's and BMPs? So here you see BMPs critical for choroid plexus fate. And this agent, CHIR, is actually a stimulator of the canonical Wnt pathway. It achieves the same result as that persistent activation reagent I told you about. So 3 micromolar CHIR and this much BMP is necessary to achieve choroid plexus differentiation. And here are these gorgeous organoids. These are human embryonic stem cell organoids. The possibilities of this are extremely stimulating, right? Because these are human cells. They can be taken from different genetic backgrounds. They can be taken from people with different disorders. Small molecules, drugs can be tested in a gene background specific manner. Cells can be reprogrammed in culture and prepared for transplantation. The possibilities of organoids are really endless. And here is our initial foray. So at day 18, it's a round ball, and then the differentiation protocol results in these blebby structures. And what's cute about it is that the blebs develop choroid markers along the periphery, okay? And comparing, so this is this red marker is TTR. It's a marker that is the defining marker for choroid plexus. And comparing mouse and organoid, you see parallels, right? The structural sort of organization of the choroid-like organization is there. And of course, we did RNA-seq to look at multiple genes. And these are all genes that are known to be expressed in choroid plexus, enriched in some, here's TTR, the defining one I mentioned. And obviously day 18, it's just an organoid, but all of these genes, when they are induced with the KIR and BMP, all of these genes go up, essentially telling us that we have achieved choroid-like differentiation. Okay, how does this connect with what I was just talking about? That the choroid plexus, when it's exposed to persistent and perpetual Wnt activation, canonical Wnt activation, can, you know, not be choroid plexus anymore and achieve hippocampal-like identity. Well, these organoids recapitulate some aspects of that also. Some aspects. So here is 3 micromolar KIR and the BMP. And here's what happens if you overdo it. If you do 12 micromolar of KIR, then you no more have choroid identity. And this is pretty clear. So this is just looking at specific genes by quantitative PCR. At day 18, it's normalized to that level. And the 3 micromolar differentiates into choroid, but then overdoing it takes it out again. Okay, so these choroid plexus molecules are definitely being suppressed in this situation. And here is the RNA-seq of that. This first set is the 3 micromolar KIR where you have choroid differentiation, and the second set is the 12 micromolar where it goes away. Okay, it goes away. Does it become hippocampus? The answer is not quite that we can pick up an RNA-seq, because see, the choroid differentiation is only along the peripheries, right? So somehow we're not able to pick up hippocampal markers in RNA-seq, possibly because of dilution, possibly because we haven't tweaked the conditions yet. But we certainly have the tidbits of a beginning. We see one hippocampal marker, and it's a defining tan hippocampal marker. So here OTX2 is a choroid marker, and you can see it's happy at 3 micromolar KIR, and then it goes down. But ZBTB20, which marks the entire hippocampus, goes up in a nice dose-dependent way. Here are the quantitations. OTX2 high at 3 micromolar and then down after that, and ZBTB creeping up. So we have reason to believe that we can recapitulate this hippocampal differentiation as well. And the future is to look at exactly how to tweak these organoids so that we can control the system better. So I'm going to now summarize. I'm going to summarize because I've talked about multiple aspects of development somewhat quickly. I see development as executing the instructions in the blueprint. Okay, developmental processes have to execute it. Evolution is actually designing the blueprint. And evolution, what is evolution, right? Evolution has to work through development. So changes in development, stretching out some developmental periods, hyperactivating some mechanisms, tamping down some other mechanisms. These are the tools of development. And these are the tools which, when modulated, can actually change the blueprint, can change the design. And that's why Evo and Devo work together. And what I've talked about today is 3 aspects of brain development. One, our studies on the wiring diagram. And I showed you how sensory nerves overshoot the subplate when LHX2 is lost, but only from subplate stem cells. And in this situation, the subplate is also electrically muted. Then I've talked about how brain structures are positioned, how the hem is an organizer for the hippocampus, and how we created multiple hippocampi by positioning the hem, extra hems in multiple places. And finally, I talked about how identity is regulated in terms of how much of a signaling molecule, such as Wnt from the hem, is required for different structures. Wnt is required to make the hippocampus, low levels of it required to make the choroid plexus, but hyperactivation of it suppresses choroid fate, and instead, at least in the mouse, induces hippocampal fate. And how we are now taking this research into organoids. Now, the organoid experiments were done at the Weizmann. We were supposed to set them up here. Arpan returned like maybe a month or 2 before the lockdown, and the pandemic put paid to all those plans. So we don't have organoids growing here yet. And I despair actually of setting them up in our sort of fungusy crumbling B block, but that's a subject for a different conversation. Right now, I would like to close with thanking all of my lab members, past and present, for this amazingly exciting ride. It has been so rewarding, so stimulating to just explore this wonderland together. And I'm really grateful for their energy and commitment and enthusiasm. And as a nod to them, today I'm wearing earrings that have data from one of my former students, Achira Roy. These are actually hippocampal neurons that she stained, and a friend made earrings which she gifted to me. So as a nod to my students and postdocs, I'd like to thank them. And I'd like to thank you for being a wonderful audience, and I will take questions.
Thank you, Shubha. Thank you very much. So I would like the audience to actually unmute themselves and let's give a warm round of applause. We absolutely have a leader in our community, and thank you, Shubha, for this wonderful talk. Questions? Questions from the audience? Please unmute yourself and ask. Actually, saw that your collaborator Upendra Bhalla had joined, I think, for us for some time. Maybe he's not here right now, but yeah. Questions? Yes, Vivek. Yes, please. Unmute yourself.
Yeah, I had a maybe very naive question. You showed the sequence of events that occurs, and so there is a timing, between these events and also an ordering of the timing. How does that happen?
That's sort of the challenge of development. I mean, everything has to be happening at the same time in a coordinated fashion. So the neural tube is forming and folding, and as it is forming and folding, already underneath it, a long line of cells has started expressing a signaling molecule called sonic hedgehog that will define its lower side from its upper side. So the back to belly axis of the spinal cord is being defined even before the neural tube has fully become a tube. So it's literally happening simultaneously.
But it happens in all cells. If you take different copies, it happens the same way. So it means this is a part of the program.
So development is an extremely robust mechanism. It's a robust program. It happens the same way in an egg of every species. And mistakes are elements. Sometimes things go wrong, you don't see them. But yeah, these basic plans, if basic morphogenesis goes wrong, the animal just aborts at some point. So in some sense, what we see as human developmental defects, the heartbreaking part is what we see is they are the more subtle defects that make it. We don't understand the main fundamental ones as well as we should. So we can't yet, broadly speaking, what makes it to birth is the more subtle versions of the major events that are, and we're still struggling understanding those. It's a challenge.
Okay.
Thank you.
Thank you, Vivek. Questions? Yeah, Shomita Mukherjee.
Yeah. Hi, Shubha. So I was just wondering, what if there are more hippocampi that are formed? What would the effect be?
Yes, can we create a hypermemory mouse? Yes. There are a few answers I can give for this one. First, it's not as if we created extra cells that made hippocampus. We, in fact, put extra signaling centers that converted what would have otherwise become some other brain structure to now become hippocampus.
Okay.
The hippocampus formed in some sense because it was forced to, because there was an extra hem next to it, but some other modality was lost. So this is a situation in which you can do a controlled experiment at all, right? Or ask, is this beneficial for the animal? So that's, that's possibly the best answer I can give.
Yeah. Thanks.
Questions? Okay, Jay Kumar has a question.
No, so there was this initial stage where something folded over and formed a little ridge, and it was in the early slides. Now, what is happening at that stage? The chemical composition, the surface tension is changing. What is causing something to fold? Because at that time, there are no muscles, nothing. I mean, where is the energy coming
Wonderful.
from?
That 30-second video of the frog egg forming a tube for the first time.
Yeah.
Actually, that is where biologists and physicists have great collaborations because it seems to, so there are no muscles, but very interestingly, there is myosin in the cells and the cells change their shape. So there is an interesting term called convergent extension, where cells physically converge, but each one extends along a particular axis. So you can change a whole pattern by each cell changing its shape, from this way to this way. And Yeah, so actually Maitreyi in DBS has some very cool work on cells literally contracting and sort of juggling with their counterparts, and has shown how myosin inside these cells locates differently from the periphery to the center when these contractions are.
But the impetus is from inside. There is no global coordinator or choreographer who does this.
No, yeah, exactly. These cells know what they're doing. Let me, okay. Our face, okay, the face that the cartilage and structure that we see, the cheekbones, the lips, the nose, all of this emerges as migrating streams from the back of the hindbrain. Okay, it's something called the neural crest. The cells migrate in streams, and that's how our whole craniofacial forms. You know, the nose, all the shapely things we see, right? Now, people wonder why two twins are similar. We should actually wonder why the two halves of our face are similar. I mean, they're not exactly identical, but by God, they're close enough. You know, I mean, imagine these cells not talking to each other, but executing instructions to give similarish cheekbones, similar nostrils, similar. My God. So yeah, there is no central coordinator. They're doing it.
Thank you.
Any further questions?
Sorry, JD, can I ask a question? This is Sandeep.
Why are you sorry? Yeah, please ask a question.
Okay. Question, Shubha, thanks for a very, very beautiful talk. And I know I've asked you these types of questions very often, and you're going to laugh at a physicist asking a spherical cow question.
He's already laughing.
Yeah, you know, this is all great, but it's so complicated. And what I always, you know, wonder is, take some really simple organism, you know, salamander, newt, I don't know, you say. Maybe you have 4 key signaling centers. It's obviously chemical gradients which are creating the signaling, chemical signaling for development. Now, is it worth asking, or are people asking the 0th order mathematical question? That is, you want so much complexity, even in a salamander or whatever to come. How many signaling centers will you need? What kind of grade? I mean, can we try to make it a little more quantitative? Is it impossible? This is biology. We just look at its beautiful data, but we give up, or what? Sorry.
Okay. Let me first try and give a slightly calming answer. It turns out that one doesn't have to tweak very much to achieve, say, a bigger brain of a human or a primate from a smaller brain of a mouse. Okay, let me try and answer it in that space. Remember I said there are stem cells and they proliferate, and then they give rise to neurons, and those neurons no more proliferate. So this means the stem cells can switch from a self-expanding mode, where one cell becomes 2 stem cells become 4 stem cells. So they increase their stem cell pool. After that, they can go into a mode where they are just in self-renewal, where one stem cell becomes 2 cells. One of them is a neuron that no more divides. The other is a copy of the stem cell. So that just maintains the stem cell pool. And then finally, the stem cell can expand itself and just become 2 neurons or 2 support cells or whatever else it's going to become. When this switch happens, appears to be the key difference between a small brain and a big brain. Okay, so it's a question of controlling when the cell cycle parameters switch from self-expansion to self-replacement to expanding oneself. And there are genetic controls on this. So it's not that scary. Okay, change from this to that. But your broader question, is there a simple system? Indeed. Where did all these genes like Wnt and LHX2 and so on, where were they first studied? They were actually studied in somewhat simpler systems. Now, the worm, C. elegans, is an excellent example of a system where, okay, this will thrill the reductionist mind. Every single cell in the worm has a name. Okay, the worm has been sectioned electron microscopically, and every cell has been labeled. The whole lineage of the worm is known from one cell to every single. The entire worm, worm specialists will correct me if I'm wrong, has something like 400-ish neurons. Each of them are labeled. So labeled means that from one worm to the other, you can find the neuron named, you know, Joe, from one worm to the other. You can laser ablate the neuron because it's a transparent system. And you can ask, what happens if you remove just this neuron? The whole worm is transparent, you zap just one neuron. Now, many insights of development have actually come from the worm. Okay, the equivalent of a signaling center is present in something called an anchor cell in the worm sort of reproductive system, where this anchor cell induces cell fate in a row of, in one cell just below it, and that cell will induce 2 other cells and so on. And you can do ablation studies and say, well, if you remove this anchor cell, will the next one become, you can do all of these experiments in the worm. So insights do come from simpler systems. But at the end of the day, you want to ask, how does the thalamus nerves come to the cortex and how do they form barrels? For that, you have to study a brain that has barrels. So there is no getting away from the complexity. But evolution has conserved the mechanisms. So you can test those mechanisms.
Okay. Thank you. Thanks a lot.
Thank you, Sandeep, for the question. And if there are no more questions, I would like to ask one question to Shubha. Shubha, you showed a diverse range of developmental studies here with a lot of imaging modality at the length scales of the entire development of the brain or small parts of the brain, right? And you mentioned that there is a control that LHX2 has an important role. You're showing clear effects of LHX2 in positioning of the subplate and how the subplate is actually put together, for example. Now, is your lab also going to look at the molecular nature of these interactions of LHX2 with different partners, which will lead to these kinds of larger length scale changes?
Thank you. You've set up actually what's going to be hopefully my next 10 years.
Okay.
So we have some fantastic data sets started by a really talented postdoc, Bhavna Muralitharan, who is a faculty member at INSTEM now. So I'm really delighted. 2 of my postdocs are faculty in academia. And Achira, whose earrings I wear, has accepted at JNCSR. So
okay.
It's great when that happens. So Bhavna triggered a whole new direction by doing the ChIP-seqs in our lab. So ChIP-seq is basically asking which gene parts of the chromatin does LHX2, the protein, bind.
Okay.
Okay, and so LHX2's endogenous protein is there, it's binding the chromatin. So the technique involves taking out this chromatin, cross-linking it so that the LHX2 remains bound to it,
right,
and then pulling the whole thing down with an LHX2 antibody.
Right.
And seeing which parts of the sequence has LHX2 bound. So we have ChIP-seq data, we have RNA-seq data, loss of LHX2 changes what. We have ChIP-seq data for cortex and hippocampus separately. Okay, because for various reasons, we know that LHX2 has different roles in both these places. We also have something called attack-seq data. Attack-seq is some parts of the chromatin are tied up and not open for readout, and some parts are open. So, does LHX2 change this open and closed nature of the chromatin? So we actually have all those data sets, and Varun, my current student in the lab, sort of heading the next generation, basically dragging me kicking and screaming into all of this quantitative data sets that I now have to learn.
Okay.
In the future, so that we know how LHX2 does what it does.
Fantastic. So maybe we're looking for another colloquium in the next 5 years. Fantastic. Thank you. Thank you, Shubha. Thank you for that wonderful talk and keeping everything on time. By the way, I typically give up to 5:15, so you don't have to worry about that much of a time. But I think that was a wonderful talk, and thank you for doing it. And I would like to thank everyone joining in today for this colloquium. And I would like to also sort of mention that next week we will have a biochemist, Professor Ruchi Anand from IIT Bombay, who is going to talk about allostery in proteins. So thank you all for joining in today. Shubha, we'll stop the